Hello, my name is Ali Akbar Setyandri, and I would like to talk to you about human blastocyst classification after in vitro fertilization using deep learning in this video. We are trying to automate the process of embryo quality after in vitro fertilization assessment by embryologists because variability among assessors remains one of the main causes of the low success rate of in vitro fertilization. Recent studies also explored the possibilities to automate embryo assessments from IVF from day 5 embryo images. In this study, we propose to do it with day 3 embryos, which only require a simple salt solution for the media. For the dataset, a total of 1084 images from 1226 embryos of 246 IVF cycles at Yasmin IVF Clinic, Jakarta, Indonesia, and we captured them by an inverted microscope at day 3 after fertilization. A team of 4 embryologists graded them 1 to 5 by using Veeck criteria, but there's only grade 1 to 3 embryos in the samples. These yields 1226 images consisting of those grades, and we pre-processed them using FastAI default preprocessor. For the methodology, we are using FastAI library using cyclical learning rates, and we fine-tune several pre-trained convolutional neural networks, such as various depths of ResNets and DenseNets, exception, and MobileNet v2. Due to the stochastic nature of the learning process, we repeated the experiments 5 times. In the table, we can see that ResNet 50 yields the best accuracy in this case, beating models with large number of parameters. Moreover, DenseNet 169 and MobileNet v2 came close. We trained these models using backpropagation with one cycle policy. From the learning curve, we can see that the loss is plateauing after the 8th epoch. Here you can see some examples of the misclassified embryo images. The best accuracy of 91.79% by ResNet 50 can even beat complex models which failed to achieve better accuracy in our conclusion. MobileNet v2, as our model of interest, to build an embedded system, achieved a similar accuracy of 91.14% compared to the best model. We argue that problems in different shades of color and digital abstractions from the image processing software might cause the misclassification. Here are some of the references we use in the study, and thank you very much for listening.